Chow Ling. Okay, probably this is a bit of revision. So I extracted the notes from Dr. Shariza. So all actually from her note, but I sort of simplified to accommodate only up to 30, 36, 35 pages or 30 slides compared to uh, her slides, which is, if I'm not mistaken, 80 or 81 pages, if I'm not mistaken. So I just take the I just took the important information that probably we can share together or we can learn together lah. Okay, because this is not my cup of tea. I only done a bit of cell culture, but never what we call it has hands on stem cell and so on. But the group of Dr. Riza and Dr. Chowdaming, Dr. Twila and Dr. Michael, so and Prof. Rosita, they are all expert in stem cell, and most of them are doing on the what we call it neuron, the brain cell and so on. Okay, probably you can plan, or you can probably you can gather an information on the topic or the subject that you might be interested to do in your final year project later on in 2 years time. Okay, so that you can gather information of each lecturer that you want to go with. Okay, probably you can start to know a bit about what their expertise lah. Okay, so we have now we have 30 plus lecturers with different kind of expertise. Some are pharmacologists, some are physiologists, some are neurologists and molecular biologists, virologists, and microbiologists, and so on. Okay, the learning objective of this class or topics is, so at the end of this lecture you should able to know, okay, or to comprehend, to the understand the basic information about stem cells and the types of stem cell and the properties of stem cell. Okay, and then relate the involvement of molecular genetic and application of stem cells. And then appreciate the therapeutic potential of stem cell via clinical trials. I think you have learned about the stem cell in your first year, if I'm not mistaken, under the topic of human anatomy and physiology. I don't know how um, they what we call assemble the topics now, because during my time we have different kind of subject. We have standalone anatomy, and then we have the standalone histology, and then we have different subject on physiology. But now, if I'm not mistaken, it all has been what we call it compiled and rearranged to become one sort of one whole story for the anatomy, histology, and physiology. Which, if you look on the organ, and then what are the physiology, the physiology of that particular organ or system, and then the histology of it. Okay, in histology or in anatomy, probably you have learned about uh, the the development, okay, from the zygote and into the embryogenesis and so on, the formation from the single cell become two and then become two times two to the power of two, blastocyst, and so on. Okay, so it's all on the stem cell. So it's all start at that particular condition where the development of what we call it undifferentiated cell start. So from that one, so it can be considered as a stem cell, and then it will be developed into a specific cell, or probably it just become a stem cell throughout their life of that particular cell. So depending on the nature of it, which is actually related with the genetic as well, as you have learned uh, in your basic genetic, where you have learned about your what we call it imprinting, the the genetic imprinting, and so on. Okay, so stem cells exist in embryo and adults. Okay, so in embryo, during the early stage of human development, or most of the animal development, either is a invertebrate or vertebrate, so it all start with sort of the same kind of nature. If you have a look on your on the fetus, the early fetus development of a human compared to probably monkey, elephant, giraffe, or so on, so it's you cannot what we call it differentiate which one is a human kind of fetus, embryo, and so on, due to the same nature of the, the physical appearance of it. Okay, once it's developed, okay, the fetus, the embryo, and then they develop into the fetus, and all the 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 limbs are being formed, okay, and into the into the sort of what we call it an adult kind of limbs throughout the pregnancy, okay, development. So then you can differentiate whether it's actually a human kind of fetus or compared to other animal kind of fetus. But early stage, it's more or less the same. So this is where what we call it uh, uh, where it's actually due to the uh, what we call it stem cell. So I have to mute a bit because I was coughing nanti kang tujuh pula you all. Okay. uh, so in embryo, stem cells function to generate new organ and tissues. Okay, so it's being sort of have their own what we call it instruction to generate whether into a new organ, new tissue, and so on. So in adults, their function to replace cells during the natural course of cell turnover spare parts. So as we grow, or as we are now currently, okay, there are some cells that that might be died off. Okay, not might be like that will die off. Probably like our skin cell, our hair cells, or whatever cell that can be that died off. Then that can be replaced or can be renewed. So it's also originated from the stem cell, but it's slightly different compared to the stem cell from the embryos, where this particular stem cell is already programmed to be whatever it's need to be. Probably the hair cell, the skin cell, the apa ni, the nail cell, whatever, the liver cell, and so on. So what is the function of stem cells in our body? So in embryo, it's the function is to generate organ and tissue because it's an early stage of life. So it's need to be developed and then to be differentiated into organ or tissue based on whatever program it's been installed onto. So in adults, it's used to repair and replace damaged or lost cells in the body. Besides naturally occurring stem cell production, they can be grown in vitro. So whatever the cells, okay, probably have done your a bit on your apa ni, tissue culture, okay, cell biology. I have been the cell that you might be play around last time, or have you ever play around with the cell, or is it all the virtual class je dengan Dr. Kumar? Dr. Choling Dr. Choling, Dr. Hanis or Dr. Seru? Ada ke? Cell biology.
Yes, doctor, we have physical practical class regarding the cell culture last semester.
Good. Good. Yeah, physical class. So some of the cells are actually uh, from the stem cell, and some are actually the stem cell itself, and it can be grown. So it will be growing into the same stem cells, and then it will not be developed into whatever it should be developed lah, because it's being sort of modified due to the nutrient that they are put in, or that they are being treated with some antibiotics and some growth hormone and so on, that actually control their life or their production. Okay, and then there are some cells that what we call it isolated, okay, from an individual, and then that being grown into cell like fibroblasts, the and some from the animal like the uh, apa CHO cell, CHO cell, the hamster cell, and then if you ever come across a cell called HELA. So it's actually from a lady, I don't know from where, probably from the Europe. Okay, she donated herself for the science lah. Okay, although she has died, I think more than 50 years ago, but her cell is still here, still being used up until now. Probably if you look around, or if you read a journal that related to cell culture, some of the experiment they use a cell called HeLa cell to 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 what we call it, to look on the toxicity, the the use of drug on the cell, and so on. You can Google it up to study the history of the HeLa cell and so on. So there are, I think there are more than 100 type of cells. We have the kidney cell, liver cell, not even plus the cancer cell and the breast cancer cell, the prostate cancer cell, and so on. Okay. So this is an overview of the how from the stem cell is being developed into uh, any one of the 220 different cells in the body. So it can be anything that is actually programmed on that particular stem cell. So which is somehow maybe related to the epigenetics as well. And then you have the kidney, or if you look on the kidney, probably you might thinking that it's only one cell, one type of cell. It's actually more than that. Okay, because kidney consists of few the skin kind of, the outer part of the skin of the kidney is from a different type of cell, although it's originated from one. Okay, and then the glomerulus, the tubule, and so on is actually from a cell, but is different from the cell with the renal cell and so on. Just like the heart cell, we have the smooth muscle cell, and then they have the elastic kind of cell that when the heart pumping the blood, okay, so the elasticity is there, and so on. Okay, and then even the brain, you have a neuron and so on lah. Okay, and in your stomach you have different kind of cell. You have the uh, squamous cell, you have the ciliated cell, and then you have the cell that have the what we call that can uh, that can secrete the 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 hydrochloric acid, gastric juice, and so on. Although it's actually a stomach kind of cell, but it's different. The upper part of the what we call it esophagus toward the lower part of the what we call it G cell, and so on lah, up until the the small intestine, the large intestine, all are different kind of cell. Although it's on the same kind of system or organ, but all are different. There are some smooth muscle cell, ciliated, non-ciliated, squamous, and keratinized for the skin cell. And then for the fingernail is something of a different, and so on. Okay, this is to show you a bit of evolution on the stem cells. So it's the study is not what we call it uh, too early, because it's probably it started in 1975, probably a bit early than that. Okay, but it's being recorded here in started in 1975. So where they study on the existence of hematopoietic stem cell demonstrated. So from hematopoietic stem cell, hema is related to the blood, probably due to the bone marrow kind of cell. Okay, as you know in your hematology, I think you have taken your hematology. Oh not, tak ambil lagi hematology.
Also the same in last semester.
Or last semester. Hematology, probably you have learned about a bit of the how the hepatocyte, the bone marrow, okay, the cell in the bone marrow differentiated into neutrophil, eosinophil, basophil, and so on. So so it's been started in roughly in 1975. To 1980, so it's the first embryonic stem cell isolated from the mouse embryo. Okay, and then the 85, roughly before 1985, the first conversion of adult rodent fibroblast into myoblast. So from the fibrous tissue into the muscle tissue. Myo is about a muscle. Okay, in 1995, so first human embryonic stem cell harvested from the blastocyst. Okay, blastocyst is at the early stage of embryogenesis. And then in 1995, I think the sheep clone from embryonic stem cell. This is the famous Dolly the sheep. Okay, the first clone sheep that was what we call it produced in the UK. I can't remember which university. I think in the Scotland, if I'm not mistaken. Okay, so and then probably at the same period, we have the leukemia demonstrated to originate from hematopoietic stem cell. And then towards 2000, uh, okay, firstly, efficient derived hematopoietic stem cell from embryonic stem cell. And then you have they start with the on the 2005, before 2005, pluripotent stem cell, and then in probably in the between 2005 and 2010, they did some clinical trial based on the embryonic stem cell to treat spinal cord injury on the patient, and so on. Although, and then towards 2015, late 2015 or early 2016, they are looking on the first trial based on the IPS. I can what as we go, I will explain about this one. Okay, so it's more about a process or a progress in stem cell. Uh, One of the major obstacle in the stem cell study, so it's about the one of it is about the ethics that if you are using a human as the as the subject to treat a disease or a problem, so they will always come with the ethical kind of concern, not just from the religion point of view, from the humanity kind of view, because you are playing, what we call it playing lah. Okay, you are testing on the living object such as a human, and so on. So although it's progressing now, but one of your senior, my student last time, I think she was graduated in 2017, if I'm not mistaken. Previously, she's working in a cryogenic kind of company in KL, okay, where there are people who are rich enough and don't know how to use don't know how to spend their money with other things. Okay, they uh, when they have their child, okay, when the baby was born, okay, their baby was born, okay, they are uh, they harvest, okay, the embryonic cell, okay, the placenta and so on from their baby, and then they store it in a cryogenic facilities in the KL, and then they have to pay, I don't know, last time I heard roughly around 2,000 ringgit per month for for the cell, okay, umbilical cord and the placenta cell to be stored or to be what we call it, keep frozen in the cryogenic kind of facilities in the KL. So now in Malaysia we do have that kind of service. Okay, so that I think was postulated by people that if the siblings of that particular, what we call it, placenta, okay, or umbilical cord tu got some genetic kind of problem, so they can use their siblings kind of a cell from the placenta and so on, okay, to treat or to go into the gene therapy and so on lah, ataupun probably apa ni, God forbidden, uh, their family got some cancer or leukemia which is related to the blood, so they can harvest or they can always refer back to that particular placenta or stem cell, okay, from their family, and then they can be used to treat cancer or leukemia and so on lah. Okay, so it's actually here in Malaysia lah, and even in Singapore I think there's much more lah. They have a very huge facilities, so for that particular thing lah. Okay, although it's still not much of a, what we call it, of a viral lah in term of the knowledge in Malaysia, but we do have that kind of facilities lah. Okay, probably you can Google it up and to find which company the there is there in Malaysia. So that's good. Okay. Okay, definition of stem cells. So it's actually undifferentiated or unspecialized cell. Stem cell is the cell that is not into a character yet. So you cannot simply say, oh, this cell is a skin cell, a brain cell, a liver cell, and so on. Like, so it's undifferentiated. So probably uh, uh, it can differentiate it after some what we call it, induction, or what's the word lah, response from the environment and so on. So the stem cell also can or can make themselves to copy themselves. Okay, they're able to make exact copies of itself. So by either self-renew indefinitely, so proliferate through cell division. So it's just proliferate through cell division is just it's mean by photocopying itself. So just like a mitosis. So they are producing the same thing all over again. Just photostate the particular cell and become themselves more. Okay, and then or either be by prolonged period in vitro culture. So it's in vitro. So it's mean it's mean that it's being put uh, into the cell culture. Okay, and then under control environment such as control FBS, the serum, and then antibiotic, the media, and so on. So that it's not being induced to differentiate or to differentiate it into a specific kind of cell. So that's what we have in the cell culture when we do cell culture. So it's actually the same kind of cell, but the difference is on the, what we call it, the word as a passage kan, PASSAGE. Probably you have learned the lower the passage is, the better the cell. So when the higher the passage, probably passage 20 or 30 and above, so the integrity of that particular cell, okay, for you to do some analysis, some treatment on the particular cell become a bit of uh, what we call it, the capacity to is not 100% anymore, probably 70% or less. So it might somehow affect your data when you do the analysis. I think we have been explained by the lecturer on this particular thing lah, the passage of the cell. Okay, or probably when you do your master or PhD with the cell culture, you will learn a lot about that one. And then the stem cell also differentiate into more specialized cells with specialized function. So where it can regenerate. So under certain physiologic or experimental condition. So under when it's become induced, and then it will what we call it change or turn into a more specialized kind of cells. Okay. Such as on the experimental condition, you can induce the cell to become a beating cell of the heart muscle, or become the insulin producing cell in the pancreas. Okay, so this is all the clinical trial, if I'm not mistaken. Okay, so undifferentiated cell is a cell that has not yet degenerated, such as or manufactured protein, characteristic of a specialized cell type. So it's not generated yet into uh, uh, the structure. Okay, the structure means the shape of the cell, whether it's squamous or what we call it, keratinized and so on. Okay, and then the protein that making up that particular cell is not being manifest yet. Okay, and then the differentiated is a cell that has acquired the features of a specialized cells, such as heart, liver, or muscle cells. So it's being differentiated. Okay. So it's either two. Okay. So it can replicate itself, a single cell that can replicate itself. Okay. So it become undifferentiated. Okay. Or it can differentiated into many types of cells. So we call it as the differentiated cells. Okay. This is sort of not characteristic, a grouping of a grouping has been done, okay, by the people, by the scientists on the type of cell or type of stem cell kind of hierarchy. If you Google around, okay, you might come across with a few kind of hierarchy. So there are some, probably only three kind of apa ni, what we call it, stem cell punya hierarchy. So there are some four, there are some even five of hierarchy. Okay, but this from Dr. Shariza, she showed uh, four, only four kind of what we call it, hierarchy. So the first one is the totipotent stem cell. So is is the ability to differentiate into cell lineage from all three germ layers. Okay, so this is at the early stage of what we call it, the endoderm, ectoderm, what we call it. Okay, the mesoderm, endoderm, and ectoderm, including placental cell. So can become the whole organism, such as zygote. So this is the totipotent stem cell. So this is probably during development from the zygote towards the uh, or way before the zygote, or towards the embryo, or the early stage of the fetus development. Okay, and then we have the pluripotent, where it's on the blastocyst kind of state. I can remember we have the blastocyst, there are more of it, right? The stage of the, what we call it, embryogenesis ni. I can remember that it's when did I learn this one? It's 19, 1999, if I'm not mistaken. 1999. It's how many years? You were not born yet at that time. Okay. Uh, so pluripotent is ability to differentiate into cells from all three germ layers with the exception of placenta, so cannot become the whole organism, such as embryonic stem cell, embryonic germ cell. So what you need to look upon the difference between all these three, okay? So for the totipotent, so it can become whole organism. For the pluripotent, so it cannot become whole organism. Okay, it's sort of going into the subspecialties of the cell. Okay, and then for the multipotent, so it's the ability to differentiate into limited number of types of from one germ layer, such as fetal stem cell, neural stem cell, blood stem cell, and mesenchymal stem cell. So, uh, from the pluripotent, so when we go into the multipotent, so it becomes a very limited. So not into more kind of specialization from the pluripotent. So it's become more specialized and more specialized. Okay, and then for the unipotent, so its ability to produce cells of their own type, such as spermatogonia or skin stem cell. So this particular unipotent stem cell is already what we call it a very specific kind of stem cell. So it's only producing only a type that is programmed to become. Let's say the skin stem cell. So they will produce only a skin cell. Okay. So probably different kind of skin cell, but it's only a skin cell in the general, because on the skin cell you know there are skin have few layers kan. You have the what we call it dermis, epidermis, and so on. The layer of the skin. So it's actually from the one stem cell that differentiated into skin cell, but probably with different kind of properties lah. But it's still a stem cell. It's still a skin cell. So this is an overview of it lah. Okay. From the zygote is called uh, totipotent. Okay. And then from the blastocyst and then epiblast, so it's is considered as pluripotent and pluripotent lah. Okay. And then toward the development of the fetus, it becomes multipotent. Okay. And then the unipotent, it become uh, when the adult stem cells, so it's either multipotent or unipotent, depending on the where it's carry out the program punya software lah, we call it program punya software. Okay. Okay, type of stem cells based on their sources. So we have the embryos, so we have the embryonic stem cells, and we have the epiblast stem cells. Their potency or their ability is a pluripotent, so it can become multipotent and towards the unipotent. Okay, so that's why most of the scientists they are looking, okay, they are looking and they study more on the embryonic stem cell. Okay, so because at the particular stage, okay, at the embryos, okay, so you can sort of manipulate, okay, the cells easily, okay, because it's still sort of still in what we call it in a naive kind of state, like it's still uh, still fresh or still apa, kalau macam let's say like a piece of paper, a very white clean piece of paper, okay, that you can do a lot of things there. You can start drawing with different kind of colors to produce whatever output that you want, unlike the on the multipotent or unipotent. So it's already being what we call has a trace on it, probably being colored by different type of color, and then you have to go along, okay, you have to accommodate whatever the color there to produce your whatever your masterpiece or your painting, but you cannot erase the color that is already there. Unlike the embryonic stem cell, is still clean. Okay, so you can draw yourself whatever you want, or you can put whatever color that you want to apa ni to to have your own artwork and so on lah. This is if you translate it into what we call it a drawing lah, ataupun analogy of it lah. Okay. And then for the adult cells, we have the bone marrow, neural, mesenchymal, cancer cell, and epidermal stem cell. Okay. For the epidermal stem cell is important lah, because it can produce into different type of stem apa ni skin stem cell. You have dermis, epidermis, and then you have the one with the keratinized and so on. The one that the cell that probably has the what we call it the secretion kind of vacuole or pocket that produce sebum, the apa ni the oil of your on your hand, on your face, and so on. If you notice, although your hand skin and your face skin, although it's more or less the same, but you can see it's different, because just look on the physical of it, the skin uh, on your hand or your arms or your leg, okay, produce more hair, rambut kan, compared to the skin on your face kan, not on that stomach lah, mustache or your punya apa ni, chin area lah for the male kan, on the nose and so on. That particular area produce more what we call it sebum kan, the oily face and so on. So it's although it's a skin cell, okay, but it's come with different kind of nature. Okay. And then for the human made from, okay, so this is the stem cell, human made from somatic cell and stem cell. So it's actually being isolated from the human, okay, and then they produce it in cell culture lah, okay. So where it's induced pluripotent stem cell, so it's become pluripotent. Okay. So now we will move deeper into a characteristic of stem cell. So we have the first one is the pluripotent stem cell. So normally pluripotent stem cell is actually the embryonic stem cells, okay, or is ESCs, okay, ESC lah, okay, embryonic stem cell. So it's the source is from the embryo, okay, either ICM or epiblast lah, the state of the embryo tu, okay. What is ICM ah? Itu dia. ICM. Itu Google juga lah. What is ICM? ICM is actually inner cell mass, okay, inner cell mass or embryoblast. Okay, so either ICM or epiblast. So this is an early stage of what we call embryogenesis. So after the fertilization, persenyawaan, okay, after the fertilization, at the age of 5 to 7 days old, okay, so it's for the during what we call it the embryogenesis. So we have the ICM or epiblast, okay. So all the IVF embryos, so apa ni, in vitro fertilization embryos. So it's if it, if it's the IVF, So it can be direct isolation. So this is where the 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 ethic or the controversial things heated up lah. Okay, because uh, so it's actually if you are taking the cell, the stem cell from the embryo, you are actually we cannot call it killing the embryo because it's not being put the soul dia punya, the life yet, although the cell what we call it apa ni, differentiate or grow. Okay, so it's not uh, what we call it kill the, because as you know, for the fetus development, and if I'm not mistaken, on the fourth month or the 120 days after the fertilization, then in Islam lah, okay, or even in sign, the soul will be put in, dimasukkan roh onto that particular fetus. Then the heart will start to beating, to beat up, and at this one on the embryo. So uh, it's will involve a bit of the ethical kind of thing lah, okay, because you actually apa ni, on purpose you are try to take or to use the particular cell from the fertilization lah, from the fertilization process. Although fertilization tu pun maksud dia persenyawaan kan, but it's not giving life yet lah. But in term of ethic tu memang, so it's not a good thing to be done lah. But probably for the sake of science or to save people's life, okay, so there are some leniency towards that particular thing lah. Okay, probably one of the alternative is to do IVF embryos lah, because you are not actually taking out the embryo, okay, the embryo, the embryo or the blastocyst, the epiblast from the womb of the organism of the mother, be it the human or mouse or rabbit or monkey and so on, when you do the IVF. Okay, so it's probably for me it's can be accepted lah. Okay, but need to have a very fine tune on it. Okay, which involve the ethics and so on. Because for the embryo, for the IVF to happen, so must have the seed or the sperm from the father, okay, and then the eggs from the mother. So it's if the father or the mother of that particular apa ni, sperm or egg give a consent, okay, probably it will be a much easier things to handle lah. Okay. So differentiation potential, it can become 220 cells. So that's why people are looking on the early as they can get on the stem cell, because from there, from one source of stem cell, they can program it, okay, or they can instruct it into to differentiate to differentiate into up to 220 cells. So it's is a very what we call it, a very good thing lah. Okay, let's say you invest 1 ringgit, at the end of the day you can produce 220 ringgit. So imagine that. So it's a very it's a very good thing lah, pakcik kaya kan. So it's very good thing to do lah. Especially if you want to treat a very rare case of congenital disease and so on. So derivatives of the three primary germ layers, ectoderm, mesoderm and endoderm lah. Okay, not the placenta. So it's very much potential of it. So characteristic for embryonic bodies or from teratoma if injected directly. Teratoma ni is a combination of different type of cell in one situation, one condition. Probably you have ever heard about the teratoma bodies, where in the stomach of individual, you found all type of cell. You found the skin cell, the hair cell, or even the tooth. Okay, some part of tooth inside segumpal. What we call segumpal in English, into to one form of like a ball. Okay, that consists of all type of cells. So it's what we call as a teratoma lah. Okay, and then for the clinical application, so it's for the allogeneic transplantation of differentiated cell or autologous transplantation for therapeutic cloning lah. Okay, probably you can Google it up. Sekarang dah pukul 9 lah. Okay, almost 1 hour, but just slide nombor berapa ni? I need to speed up. Okay. This is all computer, ada click few times dah bagus nak gerak. Okay. So characteristic of embryonic stem cell. So from the blastocyst, so you take out the stem cell, so it can be differentiated into type of cell lah. The 3 germ layers and one example of a cell type derived from each layer. So for the ectoderm, so you can produce a neuron. From the mesoderm, you can produce a blood cell. Okay, and then from the endoderm, you can produce a liver cell. So this is just like a 3 layer of what we call it blastocyst to the during the embryogenesis tu. Just like a chicken egg, kan? Chicken egg tu we have if you boil the egg, okay, you have 2 layers of it, kan? The white egg and the yellow yolk, kan? Yolk, kan? So the white tu become probably the body and the skin or the hair of the, not the hair, the feather of the chicken, and probably the yellow is become the internal organ of the chicken. So it's actually more or less like that lah. Okay, so as we have 3 layer, the first layer is can be as you probably if you can read the note down here, is ectoderm give rise to brain, spinal cord, nerve, cell, hair, skin, teeth, sensory, eyes, nose, mouth, and pigment cell. It's the ectoderm is actually on the outer layer, outer parts of our body, the skin, the hair, and so on. Okay, and then for the mesoderm, give rise to muscle, blood vessel, connective tissue, and the heart. So it's on the second layer of our body. Okay, the muscle, the circulatory system, our blood flow system, and so on. And then for the endoderm, the inner part of our body, the inner layer out of 3 layer. Okay, give rise to the gut, pancreas, stomach, lung, bladder, and germ cell, egg or sperm. So it's the inner part, the main organ of our body. So it's actually a very good kind of differentiation. Okay, so probably you have learned in your embryogenesis too, when the blastocyst form into a sort of like a spheroid, kind of a ball shape kind of nature, and then it will be folded, kan, if not mistaken, kan, yang then the development of spinal cord and so on. So this is the ectoderm lah. Okay, probably you can always refer back to your embryogenesis. Okay, so this is the embryonic body. Okay, and then this is the teratoma. When you inject the stem cell. Okay, at this time probably the teratoma is being programmed to become what kind of cell. So that's why probably if you what we call look on the internet ataupun come across on your feed or feed FEED, or you read somewhere, there's a picture of a mouse with a perfect one lobe of ear on the back of the mouse. Probably ada kot, you pernah tengok kot, I have seen that. if it's a real thing, okay, this is due to the teratoma formation. So there, the scientist has injected a stem cell that is programmed to differentiate that particular stem cell into the to produce a earlobe telinga. Probably that particular individual lost his or her earlobe, cuping telinga ni, okay, and then they isolate the stem cell, okay, probably the adult stem cell, okay, they reprogram it to produce an earlobe with the cartilage and so on. On our earlobe ni we have the cartilage that make the shape of the ear kan. If we don't have the cartilage, our ear probably be like an elephant kind of ear kan. Asyik apa? Tertutup je kan, will be covered lah. Okay. So that's a teratoma formation lah. Okay. That's one of the main what we call it major breakthrough lah in term of stem cell application and so on lah. But it's not as easy as you can see lah. It's, I think, a lot of hundred or even thousand of trial to have that kind of result. Potential application of embryonic stem cells. So for the human, regenerative medicine, such as tissue replacement after injury or disease, or fundamental studies to study of the disease model and drug screening and discovery. So where this what we call it embryonic stem cell, so you can do directly, you can do studies, you can do drug screening, you can do toxicology studies, pharmacological studies, what we call it toxicity studies on the particular cell, okay, not on the human itself. So once you got the data and it's reliable, then we can have a go, we can have a clinical trial on the particular, on the human lah. Okay, so it's unlike if you want to study the toxicity of the drug, you just inject directly to the human, so it's very unethical lah. Okay, that particular human might die due to toxicological effect and so on. But if you are using the what we call it the embryonic cell from that particular individual, or the established embryonic cell, so you might save the life of that particular individual lah. Okay, and then it will more related, or will be sort of related to the precision medicine as well lah. It can be sort of combined, or can be synergistic kind of approach with the precision medicine. And then on the animal, it can be an animal model lah, okay, to study basic fundamental and the disease development process, and then to understand mechanism behind differentiation and so on lah. Well, this is what we call it common a global kind of statement lah on the embryonic stem cell. Okay, and the potential in treating Parkinson's disease, diabetes, traumatic spinal cord injury, okay, and then Purkinje cell degeneration, Duchenne muscular dystrophy, heart disease, vision and hearing loss. Okay, so this is an example of it lah. Okay, like so on the blastocyst, so I just skip on this one lah. Okay, on the blastocyst, so you remove the inner cell mass, okay, and then this one is the endoderm, okay, endoderm or ectoderm, okay. So you produce it into a pluripotent stem cell, and then induce the pluripotent stem cell to produce a skin cell or nerve cell or pancreatic cell, and then you isolate this particular cell, so you transplant it into the patient. Okay, probably you have ever heard about the if leukemia patient okay, leukemia individual who need what we call it, bone marrow transplant. So it's more or less similar to this kind of approach lah. Okay, but normally for the bone marrow transplant, you are taking probably the bone marrow from other part of the body, or from the sibling kan, from your sibling or your mother or father or your sibling. You take a bit of it, okay, and then you transplant it into the patient. Okay, it's more or less macam ni lah, but it's not less invasive, because if you are taking the bone marrow from the individual tu, so it's actually very kind of invasive lah, sangat sakit lah. I have seen during my what we call it, LI, apa latihan industri, in my latihan industri industrial training in hospital Sultanah Aminah JB. So then at that time we were asked to go to the hospital, okay, to the patient room, okay, and then at that time the doctor want to do the what we call it, bone marrow biopsy. Dia nak ambil tulang belakang, a bit of the bone marrow lah, when the patient will be put on the bed, face down. Then they use just like a drill, okay, manual drill. They will start to poke at the spinal cord, the spinal, the vertebrae, okay, your backbone tu, okay, and then on the, if I'm not mistaken, on the lumbar, I can't remember which one, L1 or L2, L3, they and then he start to drill by hand, okay, and then you can hear the sound crack 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 on that particular bone, okay, because he use a needle to draw some bone marrow at the back of that particular patient lah, okay, to study for for bone marrow transplantation. So it's quite invasive lah, sakit lah, okay, because at that time I think there is no the GA, general anesthesia cannot be used lah, okay. So the patient is still awake and can feel the pain. Even we who are the onlookers yang stand beside him pun can feel the pain due to the sound of he try to drill with just like a screwdriver tu, okay, on the back of that particular patient lah, okay. Okay, so induced, okay, this one is another one. The first is the embryogenic. So this one the induced pluripotent stem cell, okay. So all, I think if I'm not mistaken, okay, from my opinion, the scientists, they will always targeting at the pluripotent stem cell, okay, pluripotent, so that they can play around or they can manipulate what kind of cell that they want to program to happen or to be produced. So that's why it's always on the pluripotent stem cell, okay. So it was first discovered in 2007. Do you want to proceed ataupun nak stop break? Ready? Boleh dengar?
Can proceed, doctor.
Proceed lah. Okay, proceed. Okay. Okay, so the source is from the somatic cell, okay, such as fibroblast and low potency stem cell, okay, involve no resection of embryo. So for the induced pluripotent stem cell, it will involve no embryo lah. So it's will be what we call it lenient lah, a bit on the with the ethics kind of thing lah, okay. So in this case, so the cell will be reprogrammed, okay, by using a genetic manipulation kind of process, okay, such as selective transcription factors. So all these transcription factors has been studied by the scientists. So like say the 4, okay, is uh, let me have a look Google, okay, 4 stem cell. Okay, so this is one of the transcription factors that hmm, okay, this is a marker for undifferentiated stem cell. So by using OCT4, they can control the pluripotent stem cell to become a particular or a program cell, stem cell later on, or they can use a virus, okay, and non-delivery system, okay, or by chemical reprogramming, okay, by manipulating the proteins molecule and so on lah. I think this is for your information lah, because it's quite comprehensive to what we call it to be discussed, because it's all involved about the mechanism and so on, okay. And then the culture conditions such as low oxygen, okay, and so on lah, okay. Sometimes we can control, we can play around uh, with the environment, okay, probably with the low oxygen, low carbon dioxide, or low in term of serum intake on that particular cell, so the cell will start to adapt on the different condition, okay. And then characteristic and differentiation potential, so it's once it's being programmed, okay, into a particular or specific cells, so it will more or less become similar to the embryonic stem cell, okay. So the clinical application for the induced pluripotent stem cell, so it can be more or less similar to the ESC lah, to the embryonic stem cell, okay. So this is some of the example lah to show you, okay. So comparison between ESC and iPSCs, okay. So for the ESC, it involves embryo destruction, so it will always have to do with the ethical issue. And then for the iPSCs, so it's from the somatic cell, so you can take the cell from your skin, from your liver, and so on lah, okay. So of course there will be no destruction of the embryo, and then there will be no ethical issue lah, although you still need to apply for the ethic clearance so that the authority can have a look on your method lah, to see it's in line with whatever being drawn into lah, apa yang telah dituliskan ataupun whatever being put into the law, okay. And then for the ESC, one of the advantage is it involves no reprogramming. You just directly induce the embryonic stem cell to become whatever cell that you want to be, that you want it to be. And then for the iPSCs, it's involves reprogramming, okay. So you need to program by probably the viral, non-viral, by using the transcription factors, okay, by the nutrient condition and so on, okay. So it's for the ESC is allogeneic transplantation, okay, and then autologous transplantation for therapeutic cloning. So it become uh, a bit of a common, umum, a bit of a general, okay, compared to the iPSC. So it's a disease or patient specific, okay. And then how you can produce the cell or to form a particular organ, let's say such as the earlobe on the back of the mouse, okay, by using a formation of teratoma. Both can can be sort of induced by teratoma. And then different for the ESC, it's differentiate into cells of the three primary germ layers. Yang itu lah. Ectoderm, mesoderm and endoderm. And then for the IPSCs also can be differentiated into 3 layers lah. And then efficient and more homogeneous for the ESC, then less efficient and heterogeneity, because the original source is actually from the somatic cell, and then we know the somatic cell is actually already a programmed cell. Okay, unlike the embryonic stem cell, it's not programmed yet. So this is will become less efficient lah. Okay, and then stable for the ESC, and then less for the IPSC is less stable lah, okay, due to the chromosomal instability, due to the somatic cell that you isolate for the analysis is actually an old cell compared to the ESC. Okay? So that's why if you look on the Dolly the sheep, if you read the Dolly the sheep story, so it died, I think, not too long, okay, due to when they do the postmortem, they see that the telomeres or the chromosome of the particular clone is actually similar to the donor of the cell, okay. So it's actually uh, an old cell or old chromosome. As I think if misalnya there is study, they look on the telomeres of the chromosome, okay, as we grow older, okay, the telomeres will become shorter. Let me have a look. No, telomere shortening, ada. Telomere length shorten with age. As we grow older, okay, the telomere will become shortening. Lifestyle factors such as smoking, okay. So lifestyle such as smoking, lack of physical activity, obesity, stress, exposure to pollution can increase the rate of telomere shortening. So that's why you have to be happy-go-lucky kind of individual. Okay, although we all do have stress, but don't don't stress on the small thing, and don't stress on the wrong thing. You can be stressed, but on the right kind of thing. Okay, so telomere shortening. Kalau yang so as we grow older, the telomere will be shortening. So it's happen on the dolly, the sheep. So that's why on the IPSC ni, chromosomal instability due to the telomere shortening compared to the ESC, because it is still a new, it's actually a new cell, okay, a new cell, which is probably if you look on the telomere, we compare both telomere, okay, on the same kind of cell that produced by the ESC and IPSC, okay, the same specific cell. If you look on the chromosomal, on even on the telomere, you can see the difference. For sure, you can see the difference lah. Okay. And then truly pluripotent. Truly pluripotent is a very rich in terms of manipulation. And then for the IPSC, it's not like to ISC because it contains all memories. Okay, due to epigenetic profiling, modification of the chromosome. So it's more or less similar lah, like what I mentioned earlier on the telomere shortening. Okay. And then we move on into the tissue-specific stem cells. So the adult stem cells. Okay. It should be small s here, but on this PowerPoint punya format, okay. So on the title tu, everything will be in a capital. So that's why I cannot change it. Okay. So the adult stem cell is found in various fluid, tissue, and organ. So found in small numbers. Okay. So in our body, okay, we still have the stem cell. Okay. Okay. But it's in a small number lah. Okay. So its characteristic self-renewal and differentiate. So it can become multipotent or unipotent. Okay. Properties non-tumorigenic, can directly inject into the host for transplantation. Okay, if it's non-tumorigenic. And then it stimulate paracrine effect applicable of for cell-free therapy such as scrotum, and then has homing properties as immunomodulatory properties, and then it permit autologous and allogenic transplantation. Okay, so this is the tissue specific lah. Tissue specific ni where you are isolating the stem cell from a very specific kind of organ lah. Okay, but it's depend on the type of the organ, like the brain or the kidney. There will be no stem cell for that particular organ lah, because it's the cell is what we call it obsolete. The number is obsolete. So it cannot be renewed or it cannot be multiplied. So unlike the liver and the skin and so on, so it can it can be regrown, and then can be replaced. Okay, and then I never heard the successful of the scientists who can probably regrown the brain tissue, the brain cell, or the heart cell, or even the 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 renal cell, okay, or the renal organ and so on. So that's why if people have problem with the heart, they just do heart replacement ataupun or valve replacement on the valve of the heart, and then but for the brain you cannot do brain replacement lah, because it's contain all the memories and everything. You cannot simply replace the brain of your brain with somebody brain, and then that somebody brain you want to be, you have to put you want to put all the memories, the information, the knowledge as you are before. It's not. That's why there are some blessing on that particular thing lah. Okay. So this is 1 in 10,000 to 15,000 cells in bone marrow. So hematopoietic stem cell lah. Okay. Probably in the future we might can see the development of the heart cell or the brain cell and so on lah. But I don't know if we can remove the epigenetic barrier on that particular cell. Okay, we might become successful lah. Okay, in term of producing regenerative heart cell or renal cell. But for the brain, so I'm not quite sure, because it involves something else that sometimes the science sometimes the science cannot explain. Okay, like our memory, our feeling and so on. So it's, although it's actually being involved with the hormone, okay, like the serotonin, like the what we call it adrenaline, like the what kind of hormone lagi ya, oxytocin and so on. Okay, the hormone that when they mix together, so they produce a feel good factor, a fight and flight kind of factor. When we have more what we call adrenaline, we call it adrenaline rush. So it's involved all sort of things. Okay, as for the scientists, they are very sort of, they study only this part, and then the other scientists they study only this part, and then these two scientists, when they want to connect the information together to look the thing as a whole, is quite different, because there will always be a gap or missing pieces on how you want to create a very nice jigsaw puzzle of the human physiology, especially lah. Kan? When you eat the chocolate, when you feel stressed, when you feel down, and then you eat the chocolate, especially the dark chocolate, so it produce the I can't remember the name of the hormone and then produce probably oxytocin and then serotonin to make your feel better, and then somehow on the chemical kind of nature, it vasodilate your blood vessel, okay, to make you very calm. Okay, at that time probably the adrenaline will become lower, uh, and epinephrine, whatsoever lah. Okay, so it's very very complex thing to do lah, and then to study they all, it must be in real time. So that's one big problem lah. You cannot simply cut people head, kan? You open up people head to see how the blood flow when the vasodilation occur and so on lah. So it's a very complicated lah. Okay, so susah kan, just live our life lah kan, makan tidur, kan, pergi tengok movie, okay, play game, dah kan. Okay, so example of adult stem cells. So there are few types of stem cells lah. Okay, we have the hematopoietic stem cell. So this is we are about hema. So it's all about the blood lah. Okay, so give rise to all types of blood cells such as RBC, white blood cell, lymphocyte, T, B, NK, neutrophil, basophil, eosinophil, monocyte, macrophage, and everything lah. Okay, blood related. So it's a multipotent. So from the one stem cell, so it can be differentiated, okay, into few types of cell. Okay, as listed there lah. Okay, and then you have the neural stem cell. So in the brain give rise to three major cell types. Okay, the nerve cell, the neuron, two categories of non-neuronal cell, the astrocyte and oligodendrocyte. So this is the, if I'm not mistaken, astrocyte tu yang macam what we call lah, yang shape cabang-cabang tu kan. Okay, branches kind of cell shape. Okay, and the oligodendrocyte tu is more or less the same lah, where it function as the what we call it, as a track and as a lita, okay, as a track or as a circuit, for the nerve information to be passed lah kan. I think you have learned in your physiology about the action potential and so on kan. The when and the action potential on the nerve cell tu, why is very fast. Let's say if you touch a very hot surface kan, or you touch the stove, very hot stove, okay, in a split second, in microsecond, okay, in a 1000 or 100, 1 per 1000 or 1 per 100,000 of second, your hand quickly remove yourself from touching the stove or the hot kettle and everything. So at that time it was, I think I read kat mana, it was hypothesized that the information being not just going through the circuit, is like jumping on the circuit, so it will go very fast. That's why lah. Okay, so and then they can be obtained from subventricular zone and hippocampal subgranular zone. Okay, I think this is a development that they can produce a stem cell. They can produce a nerve cell. Okay, and then it's a multipotent. Okay, but I've never seen it's being done on human yet lah. Okay, and then the epithelial stem cell. So in the lining of the digestive tract occur in deep crypts and give rise to several cell type: absorptive cell, goblet cell, Paneth cell, and endocrine cell. Okay, so epithelial ni is depend lah kan, dari goblet cell, and then you have the cell squamous, you have the enteroendocrine, because you have the endocrine that produce a sebum, a hormone, and so on. Okay, and then the skin stem cell, okay, occur in the basal layer of the epidermis at the base of the hair follicle. So this epidermis cell give rise to keratinocyte, okay, which migrate to the surface of the skin and form a protective layer. And then follicles and give rise both hair follicle and epidermis, and this is a unipotent lah. Okay, so it's very specific. Okay, key function of the adult stem cell. So they encourage repair of damaged tissue and regeneration of new healthy tissue. Okay, and then adult stem cell can facilitate the growth of new blood vessel, a process known as angiogenesis, which lead to improved blood flow in tissue. Okay, and then this cell also provide an anti-inflammatory effect, which aids in healing. So this unique combination of stem and regenerative cell present in adult cell tissue have demonstrated remarkable effectiveness in halting destructive immune response. Okay, so just read through. I think this is quite a general kind of information. The adult stem cell also aid in reducing the size of any scar tissue, okay, such as that which form after a heart attack, wound, or injury to a joint lah. Okay, probably to wound healing lah instead of using the apa we call it serum sapu-sapu kat muka tu kan, hilangkan parut-parut jerawat and so on tu. Okay, so you can do a stem cell to increase the speed of apa ni skin tissue scar and so on lah. Okay, and then last but not least, adult stem cell can prevent premature cell death. So I've got no information about this one. Probably you can Google it up lah. Okay. Prevent premature cell death, probably it will help the cell from dying off lah. Okay, from undergone necrosis or apoptosis. Okay. So advantages and limitations of adult stem cell compared to embryonic stem cell. Okay. So ethical issue, no, there will be no ethical issue. So donor or patient consent. Okay. Just if that particular patient require the cell from you, so is on your discretion lah. Kalau you nak bagi, then you bagi, you no need to go into the ethical kind of legal, okay, law and everything lah. Okay, but it's difficult to isolate and purify, and their numbers may decrease with age. So found in a minute amount, so very small. Okay, it's very hard lah. If as we can see on the presentation here, there is no information about the what we call it, the kidney cell kan. So it's probably there is no stem cell of the kidney tu, memang because it's not producing new cell of kidney and heart and so on lah. Okay, the heart tu might be okay kot because it's can be produce some from other part of the body, like probably like a muscle cell of the skin and so on. From the muscle cell tu can be lah, but from the renal tu is a bit different lah. Okay, and then heterogeneous and then genetic error. Okay, probably you can read yourself lah yang kot. Okay, because I need to speed it up, banyak lagi ni slide ni. Okay, potential application of stem cell. So the nature and function of genes that control self-renewal and differentiation processes of stem cell have made them as promising cells for various application. So there's a lot of things with the advancement in technology and so on. There will be no apa ni, what we call it, no surprise. Okay, in the future that with the technology, with a combination of technology such as the CRISPR and then gene editing. Okay, we can have if got allowed lah. Okay, we can have a very good kind of things with the health lah. Okay, for those who have probably some individual with congenital defect, genetic problem and so on. There will always a choice in treatment, such as by using the precision medicine, by look on the CRISPR, gene therapy, or even by using the stem cell. So probably people can isolate the stem cell. Okay, the stem cell that they want to put in their body. Okay, so before being put into the body, that particular stem cell can be can be treated or can be edited by using a CRISPR. Okay, to replace sequence of DNA or to add in sequence of DNA. Okay, let's say on the skin cell. Okay, probably by using CRISPR they can edit that particular stem cell. Okay, to have a protein or a gene that control what we call it melanin, no, melanin melatonin. What is the protein or hormone that make the skin color tu? Melanin, if I'm not mistaken. Okay, please recheck lah. Okay, probably the protein that control the production of melanin, because the higher the melanin kan in our body, the darker the skin we might become kan. So probably you can instead of doing what Michael Jackson did, plastic surgery, okay, you can simply do gene editing, okay, and then control the expression of melanin on that particular stem cell, and then uh, you do the cell replacement, and then your body will produce uh, slightly a fairer skin color for those who probably have problem with their skin kan, if it's too much in melanin, that probably can produce a cancer and so on. Okay, they can always go for that lah. Okay, and then for the eye color as well, okay, can probably that particular individual has some vision problem. Okay, so along the way they can do gene editing, so they can replace the eye, the cornea or whatsoever with the blue colored eye, blue colored cornea, and so on lah. Okay, you sort of like a custom made your own body, yeah. Okay, but it's a long way to go lah. But theoretically, it's not impossible lah. It can be done lah. Okay, for the stems application, okay, I think this is dekat nak habis juga lah. We have few more slide lah. Okay, uh, stem cell based therapy. So we have few here. So cell replacement therapy, or you can go for the gene therapy or cancer therapy. So we can treat the cancer stem cell or stem cell free therapy, or even the therapeutic cloning. So allow for autologous transplantation from the embryonic stem cell. And the other application that can be done is organoids, where you are producing mini organ in a dish. Okay, mini organ in a dish. Okay, it's a fundamental studies model to understand normal and disease development for the drug discovery and toxicity test. This one is uh what we call it, on the analysis. It's not on the uh direct treatment of individual lah. Okay, normally organoid, because sometimes 2D kind of cell is not what we call it, it's not enough, or it's not complete in term of study. Because sometimes, okay, when the cancer occur on a particular individual, it will attack the organ, and the organ is actually in a uh, in a what we call it 3D kind of shape. You have the 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 shape and the physical shape of the organ. Let's say uh, the what we call it, the liver. Okay, you have the liver is in a shape of a liver. If it's on the 2D, only in the what we call it, in the petri dish, you can see only 2D lah kan. You tahu 2D kan, in one layer only. You cannot see what behind it, what inside it. If they are using sort of like an organoid, okay, the mini organ, so the stem cell is actually developing into a 3D kind of shape. So with that, you can see the nature of the disease and so on. Okay, in a 3D kind of nature, you are sort of looking on an additional and on an additional perspective of the particular analysis lah. Let's say the liver organ, okay, in a petri dish, although it's not the shape of the real liver, but probably in a spheroid kind of shape, or spheroid lah, macam spheroid kind of shape. Okay, so but you can see you are adding up a different dimension instead of looking on a very flat 2D kind of nature in a petri dish. So with that, you can see how the cancer move, okay, on that particular organ, okay, and then is what we call it, an additional information that might be useful, okay, in term of fighting the cancer of that particular individual or patient lah, okay, macam tu lah, okay. Okay, so we are looking on the first one lah, okay, the stem cell based therapy ni. Okay, what is stem cell based therapy? So it's a treatment of disease based on stem cell. Okay, but what does it involve? So understanding of genes involved in differentiation process into certain lineage. So for the generation of good quality differentiated cell or pure population. So knowing the best stem cell type to differentiate into certain lineage for safe and efficient bedside application. It's actually you are targeting the right cell at the right on the right problem. So it's let me have a look. Okay. So getting a stem cell with wider differentiation spectrum that allows for safe autologous transplantation will be ideal. So understand the value available secretory molecules that are being secreted by the stem cell or stem cell differentiated cell. So this is the stem cell based chemotherapy lah. Okay. The sources can be the stem cell directly, okay, the stem cell from the differentiated cell, or the stem cell condition medium from the secretum lah, okay. So from the stem cell, so it can be differentiated into whatever the target organ that you want to have, okay. Okay, so that one is the general information about the stem cell based therapy. So this is about the one A, stem cell based replacement therapy. So what is stem cell based replacement therapy? Is actually use of stem cell or stem cell derived differentiated cell to treat diseases. Okay, so for example, treating diabetic patient. So by differentiating stem cell into a desired cells by force expression of certain genes, okay, or inhibit expression of certain genes. So, and then you can transplant the differentiated cells into the patient. So let's say, in this case, patient with diabetes is due to the pancreatic cell, okay, on the what we call it beta Langerhans, if I'm not mistaken, kan. So that cannot produce what we call it insulin, okay.